Hello students, now we will learn about auxins type of plant growth regulators or plant hormones. So auxins are first observed by Charles Darwin and Francis Darwin in canary grass tip part, which is also termed as coleoptile region of canary grass. Are first observed by Charles Darwin and Francis Darwin in coleoptile part of canary grass. Important MCQ. Coleoptile means the tissue, the name given for the tip part tissue means the tissue at tip of leaf. Now, coleoptile has a chemical that responds to light. Coleoptile has a chemical that responds to light. This responding to light, this is also used with a term called perceives. If during the question framing, if they use the term perceives also, it means that responding to. So we can give a diagrammatic representation like, if this is the tip of leaf, we can give this is coleoptile part. And this whole one has leaf. When light is exposed from this side, then this coleoptile region was bent towards that light direction. So if we see here is light, so this coleoptile part we will shade with other color. This was bent towards light. Next, after Charles Darwin, Mr. Wendt has done experiments by taking oats grass. So we will give naming as A, and this is B. Later, Fritz Warmolt, just we can write as Wendt. His name is Wendt. He first discovered and isolated auxins hormones from Avena sativa, which is also called as oats plant. Avena grass. Botanical name is Avena sativa. Common name is oats plant. So we already have seen one point that coleoptile is bending towards light. Now to do this experiment, he has taken Avena sativa grass, and we know that this tip part is coleoptile part or coleoptile region. He has cut this part. So this tip part was cut. I am representing here a separated coleoptile part. This was kept on agar-agar, which is coleoptile part. So we will write agar-agar block. We know that agar-agar is an solidifying agent. This was kept on agar block like this. Later, it was transferred to new cut Avena plant. So So this is also leaf part without coleoptile. On this was kept this agar block with coleoptile part. So this is coleoptile. This agar block which we have taken here. Which is kept with coleoptile part. Now, light was exposed from this direction. Light direction. Then, if we take here, this is light. This coleoptile part along with agar, it showed bent towards that direction. Now, we are taking this leaf little bit bent like, because it was fixed with agar block. Agar block which is kept with coleoptile region. So this will be bent coleoptile. This will be agar block. This is slightly bent leaf. So this is the experiment done by Wendt. So we have to take here, this part is Avena grass leaf. So here Avena leaf. Avena leaf. Next. Thirdly, on Auxins, Kogl. This person, he first isolated Auxins from urine from the patients who are suffering from pellagra. So, the patients who are suffering from pellagra are having deficiency of niacin, that is vitamin B3. Isolated Auxins from pellagra patient. Now, if an MCQ was asked, who first or initially isolated Auxins from humans? Even the answer will be Kogl, because patients, they are patients, but they are humans. Can you write that point also? Kogl isolated Auxins first from humans. Right, from humans. Now, this pellagra patient is deficient in vitamin niacin. This niacin is vitamin B3. So here we can take an MCQ like, if a person is deficient in niacin, then or vitamin B3, what will be the health issue? Then it will be pellagra. Next, Boyson and Joyson. These two people also did experiment to isolate Auxins. So did experiment on oats plant. Means Avena, same again. But they used mica. By using mica. Here we can see difference. By using agar block, it is Wendt. By using mica, it is Boyson and Joyson. For suppose, if this is the leaf, means oats leaf. It was cut here at this region and mica was inserted. The tip region was cut a little bit and mica was inserted. Later, light focusing direction. This is light focusing direction from this side. So what happened? The leaf has shown little bit of bent towards this direction. because light is here, and here at the tip region mica also showed some bend along with mica. So this is mica. Here along with coleoptile, that mica was also bent towards the direction of leaf. So these are the experiments done by first Charles Darwin and Francis Darwin by taking canary grass. One MCQ. Wendt and Boyson Jensen by taking oat plant, but Wendt has taken agar block, Boyson and Jensen has taken mica. So this is an MCQ. Next, Kogl, the first person to isolate auxins from humans, but the role of auxins in humans is not known till now. Role of auxins in humans is not known. So these are the isolating examples and discoveries by different people related to auxins.